Classical Mendelian genetics portrays traits as perfectly rigid. You either have yellow peas or green peas, operating like distinct, locking geometric blocks. But in nature, most phenotypic traits manifest on a smooth, continuous gradient. In the late 19th century, researchers analyzing these continuous traits noticed a strict mathematical consistency. Across distinct populations, physical characteristics predictably plot into a symmetric, bell-shaped normal distribution. Francis Galton formalized this observation through the biometrical school. By measuring thousands of individuals, he tracked ancestral heredity and identified statistical phenomena like regression to the mean, proving human variation operates on a reliable mathematical spectrum. But an opposing intellectual faction, led by William Bateson, argued the exact opposite. Relying on Mendel's newly rediscovered work, Bateson insisted that inheritance relies strictly on discrete, indivisible particles. This created a severe biological paradox. If genes are distinct, indivisible units, it seemed mathematically impossible for them to produce the smooth, blended phenotypic spectrums documented by the biometricians. Resolving this divide requires taking those continuous bell curves and proving they are the macro-level result of multiple discrete genes operating additively beneath the surface. The mathematical gap was bridged by proving that a solitary phenotypic trait is governed by the cumulative effects of two or more independent genes. This diagram details the initial breakthrough, H. Nilsson Ayle's 1909 multiple factor hypothesis. He crossed specific strains of dark red and white wheat. His resulting F2 generation segregated into predictable, distinct classes based strictly on color intensity. The mathematics proved that continuous variation emerges when discrete genes operate without dominance, stacking their effects additively. To truly understand complex traits today, examining macroscopic statistics isn't enough. We have to deconstruct the actual biochemistry of genetic additivity from the ground up. We need to start at the molecular level with enzyme kinetics, scale up to the variance of whole populations, and finally see how it all integrates into the modern omnigenic network. At the DNA level, the principle of additivity rules. Because contributed alleles lack dominance, a heterozygote produces exactly half the steady-state mRNA concentration compared to a homozygous dominant. We can observe this in the phenylpropanone pathway of wheat, where active alleles linearly upregulate the transcription factors necessary for synthesizing pigment. A 50% reduction in mRNA and enzyme levels would typically imply a severe metabolic bottleneck. Yet in polygenic systems, the physical trait does not stall. It continues to scale proportionally with the number of active alleles. This chart plots reaction velocity against substrate concentration, generating a standard Michaelis-Menten curve. We evaluate metabolic flux using the Michaelis constant, or Km. Polygenic pathways remain completely unsaturated. They reside on the extreme lower left of the curve, where substrate concentrations stay far below Km. Kasser and Burns quantified this sensitivity using metabolic control analysis, measuring a specific pathway's responsiveness through the flux control coefficient. Under their flux summation theorem, the total control within a pathway always equals exactly 1. Because polygenic networks distribute this control across numerous unsaturated enzymes, each individual gene's control coefficient remains exceptionally small, but inherently positive. In practical application, this means a 20% upregulation in one specific enzyme yields only a strict linear 2% increase in total metabolic flux. Biological lack of dominance emerges as the mechanical result of an enzyme network operating in an unsaturated first-order kinetic state. When we take that precise molecular additivity and scale it up to entire populations, we arrive back at the distinct phenotypic classes mapped out by Nilsson Ayle. Assuming zero environmental variation, you can predict the number of distinct phenotypic classes in an F2 population using a direct rule. For n independent loci, the total number of classes equals 2n plus 1. To calculate the exact number of segregating polygenes driving a continuous trait, geneticists isolate the most extreme homozygous outliers in a population and apply the extreme phenotype fraction formula, 1/4 to the nth power. In an F2 cross between 50-gram and 150-gram plants, we highlight a single extreme 50-gram outlier. 1 out of 256 offspring displaying this minimum mathematically proves 4 independent loci are involved. Across a 100-gram range, each of the 8 active alleles adds exactly 12.5 grams. The macroscopic illusion is a smooth, infinite spectrum of biological diversity. The mechanical reality is a system built on rigid, quantifiable genetic stairsteps. In 1918, Ronald A. Fisher united Mendelian genetics with biometrics. He introduced the infinitesimal model to mathematically process these endless genetic stairsteps across whole populations. Fisher invented analysis of variance, or ANOVA, to break down total phenotypic variance. He separated the genetic variance from environmental variables and partitioned the genetic total into additive, dominance, and epistatic variance. For evolutionary biologists, the critical metric is narrow sense heritability, or h². This isolates exactly how much of a population's total variance comes purely from the additive genetic component, giving us the power to predict artificial selection. It plugs directly into the breeder's equation, where the response to selection equals narrow sense heritability multiplied by the selection differential. We can calculate this using dairy cattle. If a base population averages 6,000 liters of milk per lactation and breeders select parents that average 7,500 liters, the selection differential is 1,500 liters. If offspring average 6,450 liters, the response is 450 liters. Dividing this response by the differential yields a narrow sense heritability of exactly 30%. Classical quantitative genetics treats the genome like a predictable mechanical engine, operating on the strict mathematical assumption that these independent genes do not interact with or mask each other. Treating loci as entirely independent exposes the fatal flaw in classical polygenic theory. It requires an absolute absence of epistasis. This flaw generated a modern genomics crisis known as the missing heritability paradox. When researchers mapped specific quantitative trait loci, those localized genes consistently failed to account for the total calculated heritability of complex traits. The physical reality is that intracellular networks are intensely hyperconnected. That dense interconnectivity entirely shatters the classical Mendelian idea of independent variables. To solve the paradox, Boyle, Lee, and Pritchard proposed the omnigenic model in 2017, initiating a restructuring of polygenic theory. The model categorizes genomes into 2 distinct classes. First, illuminating these large central nodes, we have a small number of core genes. These core genes are directly responsible for enacting the exact biochemical roles required to build a specific trait. The second class involves EQTLs, non-coding DNA variations mapping how sequences influence mRNA abundance. Through distant trans-EQTLs, peripheral variations alter transcription factors, creating massive energy waves flowing inward, aggressively modulating the core genes. The omnigenic model accounts for the missing heritability by demonstrating that 70 to 90. The network is so redundant and robust that it easily buffers against minor genetic variations and structural errors to maintain a stable wild-type phenotype. But the utility of this architecture is that it also allows for extreme phenotypic plasticity. Under profound environmental shifts, the network rapidly rewires its connections to produce entirely distinct physical traits from a single genotype. The desert locust demonstrates the extreme end of this capability. When physical crowding provides tactile stimulation, the locust's central nervous system floods of serotonin, entirely reprogramming its transcriptome from a solitary state to a massive, aggressive swarm filling the sky. Polygenic inheritance serves as the interconnected architectural framework that grants biological life its adaptability.